Hi friends, Namaskar. Today we are going to discuss about the batch failures, out of specification results in the pharmaceutical, how it generates batch failures or out of specification results, what are the consequences, what are the contributory factors, and how we can avoid this by the taking preventive actions. So all these things happens in the pharmaceutical. When in the pharmaceutical industry we manufacture any product, that time we have to take utmost care whatever the stages we are following. Suppose in the manufacturing stage we are manufacturing any product, and during manufacturing we have to avoid the deviations, whatever deviations. If suppose there is a weighing errors, material handling, loss of material, or equipment breakdown, manual errors, human errors, operators error, equipment error, all these are the contributory factors in the batch failure as well as the out of specification results in the pharmaceutical industry. So how we can avoid that, what is the contribution of the senior management and how it will be handled. So we will see step by step. So you have to maintain your all the means interest so that we can go proceed. During this scenario, when you are going to manufacturing any batch, that time we have to check first of all the raw materials is properly taken or not for the batch. If all the raw materials taken in the batch as per right quantity, right dosage form, and as per the master formula, then you have achieved one more step. Second step is that we have to check all the equipment instruments are properly calibrated, properly validated, and handled by the technical staff. If the equipment, instruments, and all the technique is followed, there is very less probability to find out the batch failure as well as the out of specification results. In this scenario, what happened? Third thing is that training of the manpower. If your operators, manpowers are properly trained, we can reduce lot of batch failures and out of specifications in the manufacturing industry. So training means what? The people should train on particular process what they are doing. If person is trained on particular equipment and he is handling new equipment, and in that case if he is not properly trained on that new equipment, there may be a chances to make some errors, make some any failures or some SOP deviations. Whatever the step mentioned in the standard operating procedures, that has to be followed during the manufacturing stages. Once the batch is manufactured as per the master formula with respect to the standard operating procedures, there is very less probability to find out or to found the batch failures out or the out of specification results in the batch or during the processing. Third thing, if we understand that if the formula is correct, equipment is correct, and people are trained, these are the contributions that if all the things are correct means your product is going to be in good condition. Later on, next stage is coming that the sampling of the materials. If there is a error during the sampling, maybe blend sampling, maybe tablet sampling, any contamination occurred, proper techniques are not followed, proper mask, gloves, and sanitizing techniques is not followed, there may also a probability to get the sample contaminate, and during the analysis there may be a chances for the out of specification or batch failure, because during analysis if different peak is observed in the HPLC or impurity generates, it is very difficult to identify how it is happened and what is the contributory factor. So based on the all probability or the all the tools and techniques we can do that, but on the manufacturing stage the proper control should be there and that has to be followed. Then later on, once the sampling is done with the clean equipment, clean accessories, and all the samples kept in properly in polybag, sample bag, tie it, storage is the most important factor. If suppose you have collected the sample and just put it into the drawer or just put it into the cupboard, if there is a temperature, RH is maintained or not maintained, that may be a contributory factor during your sample analysis or the your during the testing process. If that temperature and RH is not properly maintained, sample may get contaminated with the environmental condition as well as the humidity condition, temperature condition, and there may be a generate some of the impurity or degradation of the material, or the impurity generates or the loss of potency of the material. So these are the almost contributory factor during the handling of the sample. Once suppose sample is also handled properly, storage properly, then sample sent to the quality control department. During the sample receipt in the quality control department, quality control responsible to handle the sample properly, and they should keep the sample in a proper condition where the dedicated place for the sample storage, because some of the samples are more prone to the temperature and humidity condition. If the temperature and humidity condition itself in the quality control department is not followed, maybe some of the sample is cold chain sample, maybe some of the very delicate to the photosensitivity, means that sample to be kept in the dark room or the dark storage place. So all these storage conditions we have to be followed and maintained in a proper way, so your sample is going into be a good condition. Later on, in the quality control department, first of all, there are 4-5 techniques which proactively we can reduce the batch failures or the out of specification or repeat analysis activities. How it is that, like suppose if sample we are going to take for the analysis, we have to follow the method of technique or method of analysis. That method of analysis should be validated, or analytical procedure should be validated in place, and only validated technique should be used for the analytical testing procedure. Second stage, your all the glasswares, all your whatever the instruments you are using, that should be properly cleaned and maintained in a good state of condition. If suppose instrument or equipment is not properly calibrated, preventive maintenance is not done, cleaning of your vials, cleaning of your glasswares or your column is not properly maintained, that may be a contributory factor during the batch failure or out of specification results. So what precautions should take as an analyst? All the glassware should be cleaned before starting the analysis. He should ensure that all the instrument equipments are properly cleaned, preventive maintenance done, there is no calibration due, there is no breakdown, and all these things are okay. Then you have to collect the clean glassware. Later on, sample should be weighed properly as per mentioned in the your standard operating procedure or the standard testing methods, or you are in STP. Okay, so whatever sample may be 1 gram, 2 gram, properly take and weigh it, and after weighing, check there should not be any airflow on your balance, door should not be open, because if these factors are there, maybe during weighing some of the errors in the balance and wrong sample weighing may happen. So correct sample weight is the most important factor in the analytical testing. Suppose you need 1 gram, that means 1 gram, not more than or less than. Next item is that your buffer and pH, all these things should be properly maintained. The solution which you need for the analytical procedure, that should be in a proper state of condition, proper pH of that material. There should not be any error in that analytical activity or the chemicals or the balance. Next item coming that, if all these things are correct, what analyst should do? Analyst first check, is my column is correct, my column is washed, it is properly validated, it is new column, no expired of the column or life of the column is completed. All these precautions should check with respect to the column. Whatever column is mentioned in your methods or in your pharmacopoeia, that column should be particularly used, or that flow of the material or flow of the liquid will not happen properly, injection will not be done as per requirement, and there may be a bad peak shape. And when bad peak shape comes, your analysis gone west. So again you have to do the reanalysis. So that may be a contributory factor. That's why we are taking the correct column, calibrated, validated, and properly stored, cleaned column for that particular procedure. If column is good, all these things good, material weighing also done, pH or your buffer, all these things are correct, then you can run the analysis. Check the system suitability of the instrument, whether it is properly well. All these things, if you check, these are the small factors, but it will contribute more during the batch failures. All these things are okay. Check when your columns and all these you have injected, how many minutes system run is there, 60 minutes, 120 minutes, or overnight, whatever is there, make system run accordingly. Check all the audit trails. There should not be any glitches, there should not be any addition, deletion, manipulation in your audit trail. And once all these techniques followed, there is very less probability, or there is a nullification of that batch failure. Means all your batches will give a good results. After all the analysis complete, when you do the calculations, there is also one of the probability, if you select a wrong factor, wrong weight, wrong standard, or wrong something calculation factor, it may contribute into your calculation, and a whole analysis or calculation may go into the wrong direction. So calculation should be done properly. Once you done all the calculations properly, your standard weight, sample weight, blank weight, all these things are there in a good way of condition, means your all results will go into the correct direction. So if you follow all these technique tools, you can easily reduce out of specification or batch failure into your quality control department. One batch failure or one OOS will cost a lakhs of rupees, because the time intervention, chemical loss, time loss, people investigation time, then again reanalysis, then again chemical usage, all these things will be contributed, and it is a huge cost. So to avoid the cost of the company, or to control the cost of the company, as a leader, manager can contribute during the analysis. He can check meanwhile what buffer use, what media use, what is the weight of the sample, which glassware taken, is HPLC is correct, instrument calibrated, preventive maintenance done, all system suitability, tailing factor is correct. If all these things are okay, as a manager, if there is a secondary check in your laboratory, I guarantee it, there will not be any OS or batch failures. It will be minimal because of some of the times human error or something, or maybe actual problem because of the product deviations in the manufacturing stage or in the analytical stage. So to avoid all these failures, as an analyst should follow all these techniques, whatever I have told here. And if all the techniques are followed during manufacturing or during testing, you will reduce all your batch failures and out of specifications. So this is the way how the OS generates, how it will be reduced, what precautionary measures to be taken, or what contributory factors to be done. Suppose OS happened. So there are again technique to do the investigation. Phase 1, all glassware check, chemical check, means table investigation, tabletop investigation, weights, material, sample, instrument, all these checkpoints will be there. In phase 2, you have to go for the manufacturing level, cross-functional level, whether any error during the manufacturing, deviation, instrument breakdown, sampling error, whatever is there, all these things in phase 2. Resampling is there, again do the sampling by QA, resampling, reanalysis. During reanalysis, there should be a trend analyst, and you have to find out the actual cause, whatever is happened. If you got the actual cause during your probability of the investigation stage, maybe stage 1, stage 2, or phase 1, phase 2, and when these causes you identified, maybe a instrument error, manual error, people, process, method, or all these raw material things is there, we have to take a proper CAPA, and once corrective preventive action is taken, that should be implemented within 30 days. OOS should be closed, and once OOS or batch failure is closed, you have to implement the CAPA throughout your process, practices, methods and SOPs. Later on, we have to check the effectiveness of CAPA. Means suppose if you have given training to all QC analysts, but later on is there any OS happened on the same tools and technique or the method? If happened, it means your CAPA effectiveness is failure. If no OS, such type of OS within 1 year or 6 months, it means your CAPA effectiveness is in place. So that is all tools, techniques of investigation, maintenance and follow the OS practices. All these we have discussed here, stepwise, stage-wise. Hope you like. To understand all these types, you have to see the full video, understood. If any doubts, you can write us mail, comment, or if you need any next video, you can just comment it, like it, share it, subscribe it. Thank you. Namaskar.